Every time your cells divide, they receive identical copies of DNA from the mother cell. This process has occurred in all dividing cells from the time of your conception, and each cell made will pass on this exact DNA, barring any mutations that might occur. DNA replication specifically appears during synthesis, or the S phase of the cell cycle. This happens before the cell enters mitosis or meiosis. DNA replication is a semi-conservative process. In it, the original DNA divides into two components, and each original DNA acts as a template to produce the opposite or complement strand. If the original DNA reads ATTGCG, the complement strand will read TAACGC. By the time both original strands act as a template to make the new complement strand, you are then left with two copies of DNA, each made with an older strand and a newer one. DNA replication in eukaryotes and prokaryotes differs slightly due to the shape of DNA in each. In prokaryotes, there is a single circular strand of DNA, but in eukaryotes, the DNA is linear. And this creates a challenge with starting the process of DNA replication. In order to do this, there are specific sequences within eukaryotic DNA that enzymes and proteins involved in replication can identify to begin the process. These areas are called origins of replication, of which eukaryotic DNA can have several. The molecule itself is around 6 feet long. Before beginning, I want to remind you that this video is designed for non-science majors. The processes mentioned in this video is a bit of a simplification. The actual process consists of many different enzymes and proteins that I will not be mentioning here. So let's zoom in to one of these origins of replication. DNA is highly compressed and twisted, so for enzymes and proteins to access and replicate the DNA, it needs to be unwound and opened. This starts the process called initiation. Helicase is an enzyme that relieves the tension of the hydrogen bonds holding the two strands of DNA together, causing the DNA to open. DNA opens in a Y-shaped manner called the replication fork. Origins of replication appear as bubbles, with each bubble having two replication forks on either side. Helicase acts like a zipper, and without trying to sound too risque, helicase basically unzips your genes. If there are several origins of replication, eventually as DNA is copied, these origins will run into each other, eventually creating a complete copy of DNA. And once that DNA is opened through the process of initiation, elongation ensues. The enzyme DNA polymerase has an important job of adding DNA nucleotides on the opposite strand of the existing DNA strand. Recall that deoxyribose sugars have 5 carbons. The 3' and 5' carbons are important to this process as they act as directional terms. The 3' carbon is attached to the OH group, and the 5' carbon is to attach to a phosphate. DNA polymerase reads the original strand of DNA in a 3' to 5' direction, but makes new DNA in the 5' to 3' direction. That's because DNA is an anti-parallel molecule. Another example of something anti-parallel is like driving on a road where the driver on the opposite side of the road moves in the opposite direction as you. To begin the process of elongation, a primer is laid down at the beginning of replication. This primer is made of RNA nucleotides and will later be removed and replaced with DNA nucleotides. Remember that there are 2 strands of DNA to copy. One strand is replicated continuously towards the replication fork until it meets another origin of replication or at the very end of DNA. This is called the leading strand. The other strand is replicated in the opposite direction, but there is a spacing issue with the replication fork. Therefore, this strand called the lagging strand is made in pieces called Okazaki fragments. And when finished, both strands replace the RNA primer with the proper DNA nucleotides, but the Okazaki fragments leave notable gaps in the sugar phosphate background. These gaps are then filled in using an enzyme called DNA ligase. And when finished, you now have 2 copies of DNA, each made of one original strand of DNA and a new strand of DNA. Replication is complete once the leading strand reaches the end of the DNA molecule. The problem is that there is no place for the primer to be laid to allow for the final Okazaki fragment of the lagging strand to form. Due to this, one strand is longer than the other, and with the bases being unpaired on the leading strand, it causes DNA to shorten over time. To fix this problem, an enzyme called telomerase lays down telomere. The telomeres are non-coding DNA, and in humans it appears as TTA-GGG, repeated anywhere from 100 to 1,000 times. This lengthens DNA enough so that the lagging strand can be completed by DNA polymerase. Unfortunately, adult somatic cells lose telomere space with each division.
Telomere shortening is thought to be the reason why we age.
The idea being if telomerase can remain active in adults, it may reverse age-related diseases in humans and provide more longevity. If you've ever written a paper for a class, you will want to proofread it before you turn it in to ensure there's no mistakes. While DNA replication is way more accurate than you will ever do, it still makes mistakes. Sometimes the wrong base is put in place. Usually, DNA polymerase will correct it while replicating, but if the mistake persists, even afterwards, a mismatch repair mechanism is used. This system takes enzymes that cut out the mistaken DNA and replaces it with the correct one. This is called nucleotide excision repair. And one example of an important nucleotide excision repair occurs after you have been exposed to UV radiation by sunlight. UV light causes a chemical bond to occur between two adjacent thymines, causing a misshape in DNA overall. These are called thymine dimers, and if left alone, this could lead to a mutation up to and including skin cancer. Nucleotide excision repair removes the thymine dimer and replaces it with unbonded thymines. While most mistakes are corrected, occasionally they aren't, and these become mutations are permanent changes in DNA. Mutations can be harmless to very deadly, leading to certain forms of cancer. However, in gametes, a mutation could lead to a different feature on an organism. And in rare circumstances, that mutation could lead to an adaptation in a species. I mean, our opposable thumbs and massive cerebrum and the ability to walk on two legs kind of makes us freaks in the animal kingdom if you think about it. But it all began as mutations in our evolutionary history. And so now that we know how DNA is replicated so it can be passed on to daughter cells, what does DNA actually do for us? Well, in our next video, we will go see how DNA gets expressed into our genes.